The botany practicals will be of 30 marks. The 30 marks will be there in the hands of the BC students. In the botany practicals, we are dividing the question paper of the botany practicals into different questions actually. In that, the first question will be identification of the given family of a twig. Then we have to write about the floral characteristics, vegetative characteristics, technical description, floral formula, then floral diagram, then about the different identification characteristics of that particular family. It is of 6 marks. Then second question will be of the section cutting. The section cutting material will be given. We have to do the procedure of the section cutting. Then we have to make the slide of the particular thing. We have to identify which material is that. For the second question, section cutting, and by drawing the sector diagram, by performing the section cutting, we'll get nearly 6 marks. Then third question will be the experiments. Then in the botany particularly 4 live experiments will be there. The first one will be demonstration of the osmosis by the potato osmoscope method. The second one particularly will be the plasmolysis. Then third one will be the particularly the demonstration of the transpiration by the cobalt chloride method, that means leaf cobalt chloride method. And the fourth one will be the particularly chromatography, that means separation of the pigments by the paper chromatography. The 4 experiments will be there. Out of the 4, we'll get one experiment particularly. It is also particularly of what? Particularly 6 marks. Then the next thing will be spotters. The 5 spotters will be there in the botany. D, E, F, G, H on the names of these letters. The spotters will be there actually 5 spotters. 5 into 1, 5 marks. And this botany diagram is not needed. Then we have to write, we have to identification half mark, then characteristics half mark will be there particular. Then last will be of the record will be of the 5 marks particularly. Then herbarium will be of the 2 marks. Totally 30 marks will be there for the botany particularly here. And here, as we discussed now only, 6 marks for the first question, 6 marks for the 6 marks for the first question, identification of the family, 6 marks for the second question, section cutting, then 6 marks for the third question, that is particularly actually experiment, totally 18 marks. Then particularly 5 marks will be there for the spotters, that is 23 marks. Then 2 marks for the herbarium, then 5 marks for the record, particularly totally 30 marks for the botany. Then how you have to get about 30 marks? In the first question, how you have to score about totally 6 marks. In the first question, as already we said, particularly identification of the given family. The twig will be given particularly. If you are given the twig of Tephrucia purpurea, commonly called as Vempalli, or else Kajanus kajan, that is Turdal or Kandi. If these two are given, you are right about the family characteristics of the Fabaceae. Habitat, then stem, flower, androecium, gynoecium, calyx, corolla, all these things, floral formula, floral diagram, about all the characteristics you are right about Fabaceae. If the Datura metal, the white color flower, is given in the examination, you are right about the Solanaceae characteristics. If the Allium cepa or else onion is given, you are right about the Liliaceae characteristics. Remember this thing in the first question particularly, 2 marks will be there for the vegetative characteristics, 1 mark will be there for the floral characteristics. Then particularly what? 1 mark will be there for the identification, half mark will be for the floral diagram, half mark will be for the floral formula, half mark will be for the particularly twig diagram with the labeling, and half mark will be there for the lateral section of the flower diagram. Totally 6 marks will be there particular. First question. Then second question we are calling as a section cutting. That is monocot, dicot, stem, root, all these things. The material will be given. We have to cut into particularly section cutting material. We have to place on the slide, and with the help of the watch glass, slide, forceps, brush, razor blade, all these things we have to use for the section cutting. First we have to prepare the slide. For the preparation of the slide particularly 3 marks will be there. Remember the point clearly. Preparation of the slide is very important. We have to make the section cutting material into very thin, such that when you place the saffron, when you place the glycerin, and afterwards we are placing a cover slip. When you are placing the cover slip particularly, no bubble should be there on the slide or in the particular material. Then it should be very clear. And identification will be like this particularly if it is a stem. If the vascular bundles are circularly arranged, we are calling it as a U-stele. Sector diagram you have to see particularly. It is dicot stem. If the vascular bundles in the given section material, it is actually irregularly arranged, we are calling it as what? We are calling it as a attack to steel, particularly irregularly arranged vascular bundles, monocot stem. If it is root, particularly, if dicot root is there, particularly 4 xylems will be there in a particular section cutting material, that is tetra arc xylem will be there. If it is monocot root, you will be getting the poly arc xylem. That means 6 xylems will be there, particularly in your material, right? You have to show the material to the particular examiner, 3 marks. Then 2 marks for the sector diagram. Sector diagram, it means round diagram, not the lateral section diagram. Then 1 mark will be for the identification. It is second question. Then third will be about the live experiments. Always remember the point clearly. Live experiments, why we are saying in the particular botany, it means we have to exhibit that particular thing. The first one will be the demonstration of the osmosis by the potato osmoscope. Osmosis, it means the movement of the ions from the lower concentration to higher concentration with the help of the particularly semi-permeable membrane, we are calling as osmosis. With the help of the potato, take the potato, remove the peel of the potato, make it into 2 equal halves. In that potato, clearly and carefully make a dig a well, particularly so that semi-permeable membrane is created. In the potato, you have to take particularly sugar solution, that is higher concentrated solution. We have to mark particularly. Then in a beaker, we have to take the water. Then if the experiment is continued, if the osmosis is continued, actually you have to see the increase of the level in the particularly potato. If the level of the sugar solution in the potato is increased, it indicates osmosis is conducted. That is the first one, potato osmoscope, that is osmosis. And second one will be the plasmolysis. The term itself indicate plasmolysis. It means the shrinkage of the cell, particularly rheodiscolor leaves we are using. The rheodiscolor leaves, particularly, first we are taking a rheodiscolor leaves, we are making into thin pieces. Then on that rheodiscolor leaf, we are taking on the slide, particularly rheodiscolor leaves we are taking. First to the rheodiscolor leaves, we are adding the particularly water, normal water we are adding, we are observing in a microscope. Then normally we can see the cell with a benzene shape, normal cell we can see. Then afterwards, to the same leaf, if you are adding the sugar solution, higher concentrated solution, actually cell undergoes the shrinkage. You can see the plasma membrane will be undergoing the shrinkage. That is a plasmolysis that you can observe in the second experiment. The third one will be the particularly the cobalt chloride experiment, that is the transpiration. Transpiration is a phenomena in which particularly the loss of water in the form of water vapor from the plant parts will take place. In this experiment, particularly potted plant will be taken. To the potted plant, particularly the cobalt chloride paper, overnight soaked cobalt chloride paper, we are placing on a particular leaf, larger leaf lamina will be there. Then for every half an hour we are observing that particular thing. If the cobalt chloride paper, which is blue in color, if it is turning into light pink color, then it indicates the transpiration is conducted. That is third experiment. Then fourth experiment, we are calling it as a chromatography, paper chromatography. Chromatography is a phenomena, separation of the pigments on the basis of the particularly colors, we are calling as a chromatography. In that particular chromatography, here particularly in this chromatography, particularly you can see on the basis of the colors, we are separating the pigments. First, mobile phase, stationary phase, both will be there. Particularly, we are taking a beaker. In that beaker, we are taking a 9:1 solution of the petrol ether and acetone. That means 9 ml of the petrol ether and 1 ml of the acetone. We are taking a solution. It is a stationary phase. It is a mobile phase. In a stationary phase, particularly, we are taking a slide. To that slide, we are keeping it as a Wattsman filter paper. On the Wattsman filter paper, particularly on the 3/4 position, we are making a mark. On that mark, we are taking the leaf extract. The maximum we are using the spinac leaves. We are taking a mortar piston. The mortar piston, we are placing 10 to 15 leaves of the spinac leaves. Pinch of sand we are adding. Required amount of the acetone we are adding. We are grinding it with the help of the muslin cloth or velvet cloth or filter paper. We are filtering it. We are getting extract, leaf extract. That leaf extract, with the help of a dropper, we are placing on a 3/4 position on the particularly filter paper. Then that filter paper with a slide, which is called as stationary phase, we are keeping in a beaker, and such that actually the last end of the filter paper should touch the solution. The solution will travel along this filter paper. It will react with the leaf extract. Then after 2 hours, that means every half an hour you have to check. After 2 hours or 3 hours, the separation of the colors will be taking place. If green color is formed, it indicates the chlorophyll A and chlorophyll B pigments are present. If yellow color is formed, we are calling as xanthophyll. If orange color is formed, we are calling as a carotene. This is about the 4 experiments. And experiments will be of particularly 6 marks yearly. Performance will be of the 3 marks. then aim, principle, observation, inference we have to write particularly in the experiment, and the procedure is not needed, right, in the experiments. Then spotters D, E, F, G, H, 5 spotters will be there. In that 5 spotters particularly, first one will be the vegetative morphology. In the vegetative morphology, storage roots, then epiphytic roots, nodular roots, then stem tuber, culm, rhizome, bulb, then particularly what phylloid, phylloclade, all these things you are getting in a vegetative morphology. In the reproductive morphology, we will get about the hypanthodium, cyathium, verticillastra, 3 special inflorescences. Then berry, palm, pepo, hesperidium, drupe, that is the fleshy fruits, we are getting a reproductive morphology. Then about the algae and fungi. In the algae and fungi, you will get about the rhizopus, agaricus, then spirogyra. Then about the particularly the last thing will be there, that is actually see this algae and fungi. Out of the 4 slides particularly, you will get 1 slide compulsory. That means rhizopus, spirogyra, agaricus, then markantia. All these 4 you will get compulsory, 1 slide compulsory in the examination. Then about the particularly what pteridophytes and bryophytes will be there. In the pteridophytes and bryophytes particularly, we are getting about the Selaginella, Funaria, then Teres. All these things you are getting on the pteridophyta, bryophyta. In angiosperms and gymnosperms, we are getting about the Cycas megasporophyllus, Cycas microsporophyllus, then geomase, calm, that means calm plant, all these things you will get in the last angiosperms and gymnosperms. That is 5 you are getting from this particular thing, spotters. Then herbarium will be there. Herbarium and 15 to 20 particularly in herbarium will be there particularly. 2 marks will be there for the herbarium, and compulsory in the herbarium you have to remember about the botanical names, that is actually different families will be there. And that is Fabaceae, Solanaceae, Liliaceae, Malvaceae, all these things families will be there. Cajanus, Cajan, red gram, then Fasciolus mungo, Fasciolus aureus, Caesaraticum, Bengal gram, Lycopersicum esculentum tomato, then Solanum tuberosum potato, Solanum melangia brinjal, Capsicum frutensis chili, then particularly Pisum sativum pea plant. Like this, different plants will be there in the herbarium that you have to remember compulsory. Gossypium herbaceum will be particularly what? Cotton. Then Abelmoschus esculentum, lady's finger. All these things actually particularly you have to remember. And the Gingiber officinalis, particularly what? Ginger. Then Allium cepa, onion. Allium sativum, particularly garlic. Then all these things particularly fenugreek. All these things particularly you have to paste, dry and paste in the herbarium. That botanical names also you have to remember. 2 marks will be there for the herbarium. Then 5 marks will be there for the record. This is about the botany practical. No need of fear. With a confident and pleasant mind, with preparing about all these things, you will get compulsory. You will crack about 30 marks compulsory. Don't fear about the examination, and present the examination neatly. The paper should be very neat. All the presentation should be very neat. The family characteristic should be, first question will be very neat. All the characteristics should be written. And second question particularly, you draw the sector diagram. If you are in time, you can draw the particular TS also you can draw particularly, that means dicot, monocot, root or stem. And experiment also you have to write clearly about the aim, principle, observation and inference, all these things you have to write, and procedure you have to do. And spotters, in the spotters particularly 5 spotters will be there particularly. That 5 spotters you have to see clearly. 5 into 1, 5 marks. Diagram is not needed. Identification half mark, characteristics half marks. Then herbarium also you have to remember the names for this. Right? All the best. Hoping that actually compulsory you will follow these instructions and you will crack the botany practical. You will get compulsory, you will get 30 marks. All the best for your examination. Hoping for your success.